the action potential reaches the plasma membrane of the muscle cell. Now, muscle cell plasma membrane is an interesting structure. It has these membrane-lined tubules which emanate into the plasma, into the cytosol, from the surface of the plasma membrane. We call these T-tubules. So as the action potential reaches the muscle's plasma membrane, is traveling along the underside of the plasma membrane, it actually then is directed deep into the muscle itself. Their voltage-sensitive proteins mechanically open calcium channels in the sarcoplasmic reticulum. So this is kind of the large-scale view where you have the plasma membrane of the muscle cell, these T-tubules, which are membrane bounded tubules that run all the way through the sarcoplasmic reticula in a muscle cell, which is a modified form of endoplasmic reticula, is kind of flattened and surrounds these myofibrils. As the action potential comes down the membrane, goes into these T-tubules, you have this flowing sodium concentration, which means you have voltage changes along the T-tubules. Now, this is a view of what would be the back side of this T-tubule, the side that we can't actually see from this view right here. So on the back side of this T-tubule, on the T-tubule itself, you have this voltage-sensitive protein, and just opposite on the endoplasmic reticulum, you have a mechanically gated calcium channel. And when the voltage change comes along, it contacts that voltage-sensitive protein, changes its shape, it pushes up against the mechanically gated calcium channel in the SR, and calcium floods into the cytosol. And so you have thousands of these voltage-sensitive proteins and thousands of these mechanically gated channels all along the interface between the T-tubules and the sarcoplasmic reticulum. You've got lots and lots of T-tubules. The effect of this is when an action potential comes down and arrives at the muscle cell, you end up with a distribution of that sodium throughout the entire cell, so that you end up with the opening of these calcium channels nearly simultaneous across the entire cell, as opposed to a wave, which is what you might otherwise expect. So calcium channels open, calcium floods into the cytosol, all of the myofibrils as a result of that contract simultaneously. It's a very short-lived event. If you want to continue contracting muscle, the signal has to be sent repeatedly, because almost as soon as the calcium is released, it's picked up and pumped back into the SR by these ATP-driven calcium pumps. It's one of the reasons why muscle contraction is so energetically expensive. It's not just the ATP used to actually power the myosin II motor proteins, but it's also the ATP used up in the pumping back of the calcium back into the SR. So the actual interaction between calcium and the muscle is relatively simple. We have these proteins, which were not shown in the previous view, that travel the length of the actin filament, and they're typically riding in a spot that marks the position on the actin filament where the myosin IIs would normally bind. In the absence of calcium, you can add all the ATP to the muscle cell that you want. All you're going to get is these myosin II motor proteins kind of moving back and forth, but not gripping onto anything. And you get no muscle movement, no contraction. When you add the calcium, however, which is part of the process, obviously, what you end up doing is binding to the tropomyosin. It changes the position of the tropomyosin relative to the actin, and it now opens up the binding site where the myosin II is going to bind. So you need two different things. You need ATP to power the myosin II, but you also need calcium to reveal the binding site on the actin where the attachment is going to occur. All right, so that's a bit of a description of how these protein filaments function. We're now going to look at the cell division cycle, and in eukaryotic organisms, the cell division cycle relies quite heavily on the cytoskeletal elements, but we're going to talk about some other things first. The basic control of the cell division cycle, all right? I already asked this question, so we'll go on. So the cell cycle, we sometimes have the kind of inaccurate view that the cell cycle is a stop-start kind of thing, that it is somehow separated from the other things that the cell is doing. But in fact, the cell cycle, as the name really accurately implies, is a never-ending, always happening process. The whole time the cell is doing everything that we just talked about the last several weeks in regards to, copying the DNA, transcribing, translating, communicating, metabolizing, building things, delivering material to the surface to increase the size of the plasma membrane, or to change the diversity of the proteins embedded in the plasma membrane, or whatever else it's doing, converting energy, etc. These are all simply different stages of this never-ending cycle which leads towards cell division. And when two new cells come into existence as a result of cell division, the process repeats, all right? That's the only reason any of this stuff is happening, is to get to the point where we can produce two new cells from the single cell that we currently are. So it's a universal process, obviously. It's been around as long as biological life has been around. So it's this kind of series of ordered events where we duplicate the contents of the cell, and then we divide those contents into two new daughter cells. Again, it's as old as life itself, so many of the processes, certainly at the fundamental level, are the same regardless of what type of cell we're talking about. But as is the case with many of the other processes, some efficiencies have evolved and some diversions in exactly how to do it have evolved in different types of cells. Those differences are not only between organisms, but even between different cells in the same cell organism, and can actually also be different between life stages, because different requirements, when you get to the level of the multi-cell organism, put little extra demands on the behavior of cells compared to if they're individual single-cell organisms where their choices are relatively simple as to what to do and when to do it. So the fundamental stages are going to be the same for most types of cells with a few tiny differences. So first we have what we call growth 1. This is the phase that a cell finds itself in immediately following cell division. Cells come into existence with quite a bit of material already, but there is the need to kind of take an inventory of what's there and begin the process of making what you don't have in order to be able to divide yourself. So synthesis of a variety of proteins and other macromolecules. Certainly the ER and the Golgi are very busy making more plasma membrane because you need to be larger than you are when you come into existence in order to be large enough to create two new cells. Formation and growth of organelles. The binary fission of mitochondria, and if you are a photosynthetic organism, plastids. Again, in order to make sure there are enough of these things to support two new cells. There are lots and lots of biochemical switches that interact with many of the stages of protein and macromolecular growth that are able to feed back into those processes and essentially evaluate where the cell is in its path towards readiness for cell division. We then have what is referred to as zero growth, or G0 phase. Now this phase is not a mandatory phase. It's what we call an optional resting phase. There are two situations under which a cell may enter this optional resting phase. One might be if it is part of a multicellular organism, it will sit for some period of time and it will do whatever it is that it's designed to do to support the multicellular organism. Because when it comes to multicellular organism, it's the organism that reproduces to create the next generation, not the individual cells. The individual cells will reproduce to create more cells of that type, and in so doing support the goal of the organism to produce offspring for the next generation. But the cell itself isn't going to directly contribute to the next generation. So in many cases, it simply does whatever it does for some period of time. And so resting doesn't mean not doing something. It simply means not working towards cell division. It's going to take a break from moving towards cell division. The other time there might be a resting phase in a single-celled organism if the habitat, for whatever reason, is not able to keep up with the cell's needs for creating 2 new cells. There's a temporary lack of nutrients or space or something along those lines. The cell can slow things down or stop altogether for a period of time. But in a single-celled organism, as long as there's space and nutrient availability, they're full speed ahead all the time. Right? They don't ever stop and just hang out because it's kind of nice to just hang out. Right? It's only if they absolutely have no other choice because of limitations. And then we have the S phase, which is the synthesis of the new DNA. Also during S phase in eukaryotic organisms, we have a very high rate of synthesis of the proteins that are going to end up being complexed with the DNA, the histones and the other DNA-associated proteins. And then we have growth 2 phase. This is a second phase similar to G1, where the cell is now kind of taking a final inventory and seeing what few little things do we actually need to come up with in order to be ready to divide. Once a cell enters S phase, it has essentially committed to division. And so we're really just kind of looking at what is needed to complete the task. Once we finish that G2 phase, we move into the second major phase, which is mitosis, where we segregate the DNA that was copied during S phase, and then divide all of the rest of the cellular contents into 2 approximately equal halves and pinch into 2 new cells, or if it's a eukaryotic photosynthetic organism, build a new cell wall between the 2 sections of the enlarged cell. Once we finish cytokinesis, there is really no lapse in time. The completion of cytokinesis and the beginning of G1 for the 2 new cells is essentially the simultaneous event. So, again, we have these elaborate combinations of checkpoints that are made up of biochemical switches that are able to slow down or altogether halt things, processes, depending on what is going on, what types of extracellular or intracellular signals are being received. So perhaps a process has not completed the way it was supposed to, some sort of negative feedback perhaps. So let's say you're synthesizing a whole bunch of proteins that require lysine. The lysine is part of a negative feedback that feeds into another process. And if there's a lot of lysine, that's maybe an indirect way of knowing that the process that was supposed to consume lysine hasn't yet really finished. And so the lysine is holding the next stage off just a little bit. That's just a simple example. There would be hundreds or thousands of similar types of potential feedbacks. So it could be something wrong with the cell. It could be that maybe the enzyme that is in charge of a particular biochemical pathway is for whatever reason defective, and you're never going to get that negative feedback loop to correct itself. Or it could just be something relatively temporary, lack of nutrients or something like that. Again, in either case, we could have a stoppage that is either temporary or permanent. Temporary in the case of environmental deficiency that could become permanent if the environment never actually ends up getting to the point where it can continue the process. Typically, something wrong with the cell is going to lead to a permanent stop, and that cell division process is never going to happen. That cell line is going to simply disappear from existence. We talk a lot so far in here about the cell's ability to kind of fine tune the rate at which processes proceed. We used an analogy a couple times of like a dimmer switch, where things are rarely fully on or fully off, but rather somewhere in between. It's a little different when it comes to the cell cycle. In the cell cycle, we actually see something that's kind of more like a rocket launch, where everything is in order, everything is ready, or it isn't. And if everything isn't ready, if you've only copied 98% of your DNA, we can't go forward with the next step. So in the case of cell division cycle, a very small amount of negative signaling is generally sufficient to slow down or even completely halt progression to the next phase of the cell cycle. So this whole system is based on the function of a series of biochemical switches, very elaborate biochemical switches that interact with each other in very efficient but complex ways. This system is based on a family of kinases that are cyclically activated. In other words, they are activated during different parts of the cell cycle. So we call them cyclically activated cell cycle kinases. Now, a kinase is simply a protein that phosphorylates a target protein. The cell cycle kinases are kinases that phosphorylate target proteins. It just so happens that the types of proteins they phosphorylate are proteins involved in some aspect of the cell cycle. Now, cyclically activated means that they are only able to phosphorylate their target if they are activated, and that activation event occurs at very regular intervals throughout the cell cycle. So there are lots of different types of cyclically activated kinases, each of which phosphorylates a different protein. And these different proteins are simply involved in aspects of the cell cycle and are able to control different elements that are part of the overall progression towards cell division. When we think about the cyclically activated kinases, we really just need to imagine how many different things have to happen in order for a cell to divide. And the answer is almost everything that a cell does is in some way related to its movement towards a point in time where it is able and ready to divide. So these target proteins, many of them are transcription factors. Many of them are proteins that interact with the ER and lead to the formation of budded, coated regions of membrane, eventually budding off and traveling to the plasma membrane. And the list goes on and on. So they can initiate a cell cycle process. Some of them inhibit a cell cycle process, and some simply modify a cell cycle process. So the fundamental connection is when the kinase is activated, its target protein is phosphorylated, and when its target protein is phosphorylated, something related to cell cycle happens. So the activity of these kinases is controlled through regulatory ligand binding. And in the case of these types of kinases, the regulatory ligand that controls their activity is a small protein, all right? A small protein. So just to kind of back up and remind ourselves of what kind of activity we're talking about, you have a kinase, and it's sitting there, and it's inactive. But at some point, its regulatory ligand, which happens to be a small protein, shows up on the scene and binds. And what's nice about the regulatory ligand being a small protein is that we can control when that small regulatory ligand shows up by controlling the transcription of that protein. So we transcribe this protein. This protein happens to be a regulatory ligand. It binds to this kinase, this kinase now acquires activity, and it's now able to go phosphorylate its target. And its target might be, a protein that is involved in some metabolic pathway that is necessary for some aspect of cell division, right? And again, there are thousands of potential targets for these particular types of kinases. So these little regulatory proteins that are able to activate some of these types of kinases are referred to as kinase activating proteins, all right? There are a number of different kinds, but by far the most common and the most important is a family of regulatory proteins called cyclins, all right? Cyclins are a large family of small protein that function as the regulatory ligand for this category of kinase, which is involved in controlling cell cycle events. The kinases whose activity is dependent upon this family of small regulatory protein ligand are called cyclin-dependent kinases, or CDKs for short. All right? Cyclically activated kinases whose activity requires binding with the regulatory ligand cyclin before their final target can be phosphorylated. So lots of different kinds of cyclins. Most cyclins are able to bind and regulate the activity of more than one type of kinase because often a particular event requires the coordinated activity of several different kinds of proteins. And so we can introduce a single type of cyclin. It can go and bind to 20 different kinases, which can go off and phosphorylate 20 different targets, and all at once we get 20 different proteins, each doing the thing they do as part of a big event. So just imagine the copying of the DNA, how many different proteins are required for that? And you don't want to have to go kind of pick and choose and turn them all on individually one by one. You simply have a single type of regulatory ligand that effectively turns them all on at the same time. Also, some CDKs are able to bind more than one type of cyclin. So I'm a cyclin-dependent kinase, and I bind this particular cyclin, and I adopt a conformation that allows me to phosphorylate this target. At some point later on, I may bind a different cyclin, adopt a different shape, and phosphorylate a different target. So the same CDK may have more than one target, and which of the targets it ends up phosphorylating depends on which type of cyclin has been delivered to the scene. So we've got a lot of versatility in the mixing and matching of these CDKs and their cyclins. The cyclins themselves undergo cyclical synthesis and degradation throughout the cell cycle. So it is the production of these different types of cyclins that is most fundamentally in control of the activity of these cyclin-dependent kinases and the, therefore, activity of their targets. The CDK levels, the level of the CDKs themselves remains essentially constant throughout the cell cycle. It is the changing concentration of the regulatory ligand, the small cyclin proteins, that changes throughout the cell cycle to change what's going on. So the cell has a lot of things that it has to be concerned with in regards to cell cycle. And what does evolution like to do when it has something that seems to be working pretty well? What's the very next thing evolution likes to do?
If it's working well, leave it alone.
No, it never does that. If that were the case, we'd have 5 proteins and we would have one day of cell biology, and that would be that.
Shouldn't make it complicated.
Not complicated, that's how we interpret it.
Make it more efficient.
Make it more efficient. Evolution is annoying in that it is never satisfied with what it has. That's why we end up with what we perceive as complexity. We know we only need 450 proteins to survive. Mycoplasma genitalium shows us that. It's not about surviving, it's about evolution's never-ending drive to make it just a teensy bit better. And how long has evolution been working on cell cycle?
Forever.
Well, as long as anything it's been working on, right? I don't think forever, but maybe. We really don't know. A very long time. That's my nice way of saying it's about to get even more complicated. So there are lots of additional proteins that further modify this already somewhat complicated relationship between cyclins and CDKs. And again, the reason is because it's not only critical and complex, but evolution has been tweaking it for a very, very long time. So one of these additional proteins is something called CDK activating kinase. Turns out that when the CDK and the cyclin first come together, the cyclin-dependent kinase still isn't in a conformation whereby it can phosphorylate its target. CDK activating kinase is a separate kinase that phosphorylates the ATP-bound CDK, preparing it or getting it ready to be able to actually do its thing. We also have inhibitory proteins that can take an otherwise ready-to-go kinase and at least temporarily prevent it from going forward. So we have our CDK. It's there, it's not doing anything. It picks up its regulatory ligand. It now adopts a conformation where it is getting close to being able to target, phosphorylate its target, but it's still not ready yet. What we actually need is another element, a cyclin-dependent kinase activating kinase to come in and phosphorylate this structure, creating a conformation in which the kinase is now able to phosphorylate its target. Even if we have a fully active structure, a fully active complex, they're not showing the ATP here, they're just showing the activating phosphate. We still might temporarily wrap it up with this inhibitory protein. There's even more control, however. There's another kinase called WEE1 that potentially can add another phosphate to the CDK, which is inhibitory, and a phosphatase that would then be needed to remove that inhibitory phosphate in order to reestablish the activity of the CDK. So the complete picture, if you will, looks something like this. We have a CDK. It binds its cyclin. The CDK activating kinase comes in and adds the activating phosphate. This thing is now ready to go. However, we often will see that before it has a chance to do what it does, this WEE1 kinase will add an inhibitory phosphate. And before we can get back to our active state, we need CDC25 phosphatase to remove the inhibitory phosphate. Now we're going to look a little bit later on, we won't get to it today, but on Wednesday, we will look a little bit at exactly what the benefit of this extra level of control is, right? And there is a benefit, and it has something to do with getting a whole bunch of CDKs ready to go, like an army, which takes a little bit of time, and adding inhibitory features as you build your army, and then introducing a signal that removes that negative control very, very quickly so that the entire army is unleashed at the same time. And we're going to look at exactly how that is related to replication as an example. So in order to activate the CDK, you need appropriate levels of the cyclin, which is the regulatory ligand in all of this. If you do have your level of cyclin, you'll get your cyclin CDK complex to form. You then need the CDK activating kinase to add the activated phosphate to that complex. Then you either have to have no inhibitory phosphorylation by WEE1, or if you do, you need to have that inhibitory phosphate removed by the CDC25 phosphatase. And then finally, superimposed upon all of this there can't be any of these inhibitory proteins masking the active site on the cyclin CDK complex. All right, and if all of these things are in place, then and only then do you get your target protein phosphorylated, right? Just very quickly, control of cyclin levels is really just a balance between the synthesis of the cyclins and their degradation during different regions, different periods of the cell cycle. So we have transcriptional control over cyclin synthesis, and we have ubiquitin ligases, which are always on the control for cyclins, and different signals that they pick up following their incorporation into a complex will attract those ubiquitin ligases. It's so kind of just a little thing to keep in mind. It's the level of any particular regulatory cyclin that goes up and goes down throughout the cell cycle, which is why they're called cyclin. Kinase levels remain constant. Cyclin levels go up and down, which means the activity of a kinase goes up and down. But the kinase itself level is the same, right? And these are kind of, I'll say interesting. Cyclins all behave, their increase and decrease in concentrations all follow sign functions. For every cyclin, there is a specific sine wave function that very, very accurately depicts the rise and fall of their concentration, which illustrates, among other things, that their increase and decrease is not random or arbitrary or just kind of whatever. Very, very precisely controlled. When a cyclin has a sine wave function assigned to it, when they discover what that function is and they assign it, it's the same from one cell to the other, cycle after cycle. All right, so we'll end it there for today. We'll finish up with a little bit more